Hello and welcome to this video. I'm Tristan from Math Against Malaria. We are an enthusiastic research group that works at the interface of infectious diseases, mathematical modeling, biostatistics, bioinformatics, and genetics. In today's video, we'll cover how malaria transmission works in humans. So let's get started. Let's start by reminding ourselves what malaria is. Malaria is one of the most relevant infectious diseases. It causes worldwide over 230 million infections and 590,000 deaths every year, according to the latest estimate from 2021. The pathogen that causes malaria are parasites of genus Plasmodium, which we see here under a microscopic image as this characteristic ring stage forms. Plasmodia are unicellular organism, meaning each parasite consists just of a single cell. They are haploid, which means that in their genome, they have only one copy of each chromosome, like, for instance, bacteria do. However, unlike bacteria, they are eukaryotes, which means that they already have a nucleus, like our human cells, which makes them a higher developed organism. Malaria is not just a human disease. It can infect a number of animals too, including other mammals, reptiles, or avians. There are 5 species of Plasmodium which frequently cause malaria in humans. The most relevant and most dangerous by far is Plasmodium falciparum, responsible for most infections. Other species are Plasmodium vivax, Plasmodium ovale, which are 2 species actually, Plasmodium malaria, Plasmodium knowlesi, and there have been rare reports of other Plasmodium species infecting humans too, including Plasmodium cynomology, Plasmodium innoi, Plasmodium brasilianum, or Plasmodium simium. Let's start to describe the transmission cycle of Plasmodium falciparum. For malaria transmission, we need humans and we need female Anopheles mosquitoes. There are about 100 known Anopheles species which can transmit malaria to humans. Female mosquitoes need to take blood meals because they need the protein from the blood in order to hatch their eggs. If our malaria transmission from mosquito to human occurs, the mosquito inoculates sporozoites from her salivary glands into the human body, and this starts the exoerythrocytic cycle. Sporozoites will find their way into the liver, where they invade liver cells and mature into schizonts, which will finally rupture and release parasites into the bloodstream in the form of merozoites, which ends the exoerythrocytic cycle and starts the erythrocytic cycle. Here, merozoites infect red blood cells and first form immature trophozoites, which appear under the microscope as the characteristic ring stages, and then further mature into fully grown trophozoites that further grow into schizonts, which ultimately rupture and release new merozoites into the bloodstream, which will again attach to new red blood cells and perpetuate the cycle. One of these cycles takes approximately 48 hours or 2 days, which causes the characteristic fever cycles of malaria. Importantly, all these parasites so far are asexual and cannot transmit malaria from humans to mosquitoes. For human to mosquito transmission, Some of the merozoites have to differentiate into so-called gametocytes, which are sexual forms of the parasites. They don't reproduce anymore in the human body or change the pathogenesis, but they are the key to human to mosquito malaria transmission. Namely, if a new mosquito comes and takes her blood meal at an infected host and during that blood meal ingests male and female gametocytes in her gut, the gametocytes will immediately release gametes, i.e. sperm and egg cells, which immediately fertilize and form zygotes. These zygotes will grow into ookinetes, which can penetrate and dig the mosquito's midgut wall and dig a hole through it. The ookinete will grow into oocysts, which further grow, rupture, and release sporozoites, which find their way into the mosquito's salivary glands. And now this mosquito can infect new humans, and this closes the malaria transmission cycle. Other human pathogenic malaria species have a very similar transmission cycle. However, their transmission cycles have some particular characteristics which we should discuss now. So let's start with Plasmodium vivax and Plasmodium ovale. A distinct feature of these two species is that dormant parasites remain in the liver. These parasites are called hypnozoites. Even if all parasites are cleared from the blood, hypnozoites can be reactivated weeks, months, or even years after the original infection and start the erythrocytic cycle again to cause a new symptomatic infection. In Plasmodium malaria, hypnozoites do not exist. However, some of the blood stage parasites also fall into dormant stages, which might be activated after years after all the initial infection was cleared and cause a recrudescence of the infection. A distinct feature of Plasmodium knowlesi is that it has a non-human primate transmission reservoir. So transmission does not just occur between mosquito and humans, but also between mosquitoes and non-human primates. This concludes how transmission in malaria works. If you liked the video, we would be happy if you give us a thumb up and subscribe even to the channel. If you want to read more about how to model malaria mathematically, please check out some of the publications that are linked below. Thanks so much and see you next time. Bye.